Hi everyone, thank you for your interest in our poster entitled Hybridization LC-MS, a selective solution for PMO and PPMO bioanalysis, presented at the TIDES 2026. My name is Mingluan Chen. I am Principal Research Scientist in LC-MS Mass Development Group at Altasciences, and it will be my pleasure to discuss our research with you today. PPMO and the peptide conjugated PMO, known as PMOs, are emerging therapeutics approach to support PK evaluation. Sensitive and selective bioanalytical methods are essential. However, unlike conventional oligonucleotides, which are highly selectively charged, PMO is charge neutral, while PPMO contains a peptide conjugate that can introduce positive charges. As a result, these compose behavior differently in LC-MS analysis, which complicate both extraction and detections. Given these challenges, a more selective and robust analytical approach is required. The objective of this work was to develop the hybridization LC-MS method for the selective and reliable quantitation of PMO and PPMO in biological matrices. In addition, we aim to optimize hybridization extraction parameters to improve selectivity and recovery. To achieve this, we developed a workflow combining hybridization extraction with LC-MS analysis. As shown in Figure 1, rapid plasma samples were subjected to hybridization extraction using a biotinylated capture probe. The probe analyzed complex was conjugated to a stratavidin magnetic bead, followed by washing steps to remove matrices interferences. The analytes were then thermally eluted and prepared for LC-MS analysis. Key parameters, including the probe chemistry, buffer composition, and the probe-to-beads ratio, were systematically optimized to improve extraction efficiency. Following extraction, chromatographic separation was performed using a reverse phase LC system coupled with SAIX 6500 plus triple quadruple mass spectrometer, operating in positively ASI mRNA mode. Detailed LC-MS conditions are optimized in Table 1. With the method established, we then evaluate its performance. We first evaluate capture probe chemistry. As shown in Figure 2, DNA capture probe showed significantly higher single intensity compared to the PNA probe, indicating better hybridization efficiency and recovery. Based on this result, the DNA capture probe was selected for following mass development and qualification. Next, we evaluate chromatographic conditions. As shown in Figure 3, the use of TFA in the mobile phase improved the peak shape and the chromatographic resolution. However, it's also introduced ionization suppression, leading to reduced single intensity. To address this, post-colon addition of the ammonium bicarbonate was used, which significantly improved single intensity. Under optimized conditions, as shown in Figure 4, PMO and PPMO under the internal standard were well resolved, with sharp shape profile and the consistent retention times. It is important to note that despite the positive charge introduced by the peptide conjugate, PPMO also show a well-defined peak shape. This enables reliable detection and quantitation at low concentration levels. The method was successfully qualified over the calibration range of 2 to 1,000 nanogram per ml for PMO and 5 to 2,500 nanogram per ml for PPMO. As summarized in Table 2, the method demonstrated good accuracy and precision across all QC levels. For PMO, the accuracy ranged from 97.7% to 101.5% with precision below 6%. For PPMO, precision ranged from 97.9 to 103.5% with the precision below 9%. This result indicates reliable and consistent performance across the calibration range. In addition, as shown in Table 3, evaluation across multiple matrices slot, including the individual donors and the hemolyzed plasma, showed consistent results. No significant matrix effect was observed as measured concentration across different matrix lots remained within acceptance criteria. Overall, these results support the reliability of the method for quantitative bioanalysis in complex biological matrices. Based on this result, we can now move to the overall conclusion of this work. In conclusion, a hybridization LC-MS-MS method was successfully developed and qualified for the quantitation of PMO and the PPMO in rabbit plasma. The method provides enhanced selectivity through the target extraction, along with the improved chromatographic performance and the sensitivity. This approach is well suited to support the bioanalysis of the PMO-based therapeutics and another complex oligonucleotide approaches. And thank you very much for your attention, and please do not hesitate to reach out if you have any questions.